Shall we start with a sound check? Can you hear me in the back there? Loud enough? Am I loud enough there in the back? Excellent. Wonderful. I'd like to start with a few course matters, just to let you know about some of the things that we have available for you to help you master the material. Susan will be has organized a review session for Tuesday, this coming Tuesday, 6 to 8, and it'll be held right here. So that's a convenience. You don't have to try to find something in Bowen, which is, we have to give people, balls of string and birdseed and stuff to find out the way out. I think Angie's hoping to set up one for Wednesday, so if Tuesday doesn't work for you, there may be one on Wednesday as well. On Icon, after the class, you're going to be able to download, if you want to, a practice exam from previous years. There's a list of tutors available there if you want to get in touch with a biochemistry major for tutoring. Some of the slideshows I've had before class as sort of the ambience and stuff, if you want to see those in more detail, they can be downloaded. There are more articles listed under the articles section for those of you who want to follow up on any topics. The things in the articles will not themselves be the subjects on the test. Those are completely complementary, supplementary material for people who want to go further. Under the links, there are links to Google Video, which has each of the class lectures so far available in the Google format where you can jump to the part of the lecture you need. And there are also now several links to online textbooks. Sometimes when people are stuck with a concept in the textbook you're using, it helps to look at it in another textbook, and this way you don't have to buy them. I'm interested, if anybody does use those textbooks to have feedback on them, it is conceivable that we could simply have an online textbook next year, and I would want to know that the textbooks actually were equally good as what we have. I doubt they will be, but maybe they are, and please let me know if you think that's true. Susan has asked me to remind you that in addition to the PowerPoint slides available, there's a list of study questions available down here. Are there any questions then about protein structure, protein folding, hemoglobin, myoglobin from Monday and Wednesday? Okay, well, let's press on then. This was open a moment ago. There we go. All right. So what we're going to be talking about today in one lecture is the cytoskeleton and other structural proteins. Don't understand why it takes these things so long to load, but there it is. The cytoskeleton is a component of the cell that has really been growing in its appreciation in the last couple of decades or so. For a while, it was not even thought that there was much of anything in cells, because if you don't have a way of analyzing it or isolating it, it's hard to get a handle on it. We've come to appreciate that, in fact, the cytoskeleton is a very significant part of the cell. It can comprise anywhere from 25 to 80% of the protein in the cell. So just on a mass basis alone, it's very important. It's the cytoskeleton that provides shape to cells. So when you look at a cell and you see the shape of a neuron, or you look at the cell and you see the shape of a lymphocyte, what you're seeing are neuron cytoskeletons and lymphocyte cytoskeletons. The cytoskeleton is the basis for movement for most cells, most eukaryotic cells. This is a strictly eukaryotic phenomenon we're talking about now, although we can get into its roots if you want to. The most important kind of cell movement, of course, is cell division. If for cells to grow, you have to have some way of turning into two in most cases, and the cytoskeleton is responsible for that. Some cells are capable of swimming with cilia and flagella, and that's mediated through the cytoskeleton. Even more cells are capable of crawling. So if you have a wound in your body, the epithelium and the fibroblasts literally crawl in to fill in that wound. The cytoskeleton is what confers mechanical strength on our bodies. A very dramatic example is people who have a very small alteration in the cytoskeleton of their epithelium develop a disease called bullous pemphigoid, where just getting born causes blisters to form. The skin is so delicate. I've mentioned how the shape of a cell is a differential characteristic between cells, but a number of aspects of the cytoskeleton are different from one tissue to another, so that when you look at a differentiated phenotype, anywhere from, again, 20 to 80% of that phenotype is a cytoskeletal phenotype. Cancer cells have awful cytoskeletons. In fact, pathologists can tell if they're looking at a cancer cell or a normal cell because of the disorder in the cytoskeleton. That you can, in many cases, tell by looking at it that it is a cancer cell, and that's because of changes there. This particular set of pictures is one I think that helps bring across the idea that there is a cytoskeleton in the sense of an integrated, cross-connected, coordinated system rather than three separate systems in the cell. The red here is actin, the blue here are microtubules, and this is intermediate filaments. But these are all the same cell. And you can see how the intermediate filaments and the microtubules are more related to each other. You can see how the actin filaments are kind of providing a base plate for the rest of them to be related to. But then if you're careful, you can see that many of those actin filaments are places where microtubules terminate. So it's not a random collection of pieces. It's a closely integrated system. Let's talk, first of all, about one of the great systems in the cytoskeleton, the first that was extensively described, which are microfilaments. These are filaments that are about 7 nanometers across. They are often found underneath a cell membrane. If you look at the cell membrane, there'll be like a felt of thin filaments or actin filaments there. In some cells, they form bundles or cables. If you remember this cell, this is not an individual microfilament, but this is dozens of them bundled together. And the individual proteins that make up microfilaments are the protein actin. Here's a single globular protein of actin. So it's a single polypeptide that's the building block. If you look at this carefully, you can get a sense that there's a twistiness to it. It's a twofold helix of the subunits packing together in a kind of twisting way. The twist is a little bit clearer in this reconstruction that's been done from electron micrographs. Inside of the actin, you can see here this green bit. That's going to be either ATP or ADP. So it binds adenine nucleotides. And we're going to see throughout the semester that many proteins bind adenine nucleotides for lots of reasons. They tend to be either driving energy or providing information or carrying groups, all kinds of interesting things. And so actin is one of these ones that has ATP or ADP bound to it. When actin polymerizes, we call it F-actin for fibrillar actin or fibrous actin. When it's as free individual proteins, we call it G-actin because it's the globular monomer. And so it's the building block for one of these three systems. Here you can see the helical character a little more easily because it's in a diagram. And what's not more interesting, but what's also important about this helical structure that's made up of the monomers is that it is polar. The two ends of the filament are not the same. If we start with a little sort of small subunit of a nucleus, that nucleus of 2 or 3 subunits does not form very quickly. We can make artificial nuclei by shearing long filaments into short filaments. But once there is a nucleus of some kind or other, elongation happens more quickly. And so you can have these globular subunits add to the filament and make it longer quite quickly. That's polymerization, making a polymer. Now this is not a covalent polymer. This is a non-covalent polymer, but it's a collection of many, many globular actin monomers. One of the interesting things about this polymerization process is it doesn't happen the same at both ends. So one end is assembling more quickly than the other. And you can see that here by having a large number of the new units added compared to a small number here. And that difference holds up over time so that you can tell that one end is assembling less rapidly than the other. Once these globular actin subunits have assembled into that polymer, the ATP is hydrolyzed. into ADP. There's always an adenine nucleotide bound, but it's ATP in the free form and is hydrolyzed during polymerization. This goes on. It's happening at different rates at the different ends because they have different affinities for the actin. And if you think about this, this would suggest that polymerization will continue until all the subunits are in the filaments at equilibrium. But since the fast assembly end has a higher affinity than the slow assembly end, you can be adding monomers at one end and losing them at the other, and that would be a steady state. It's not an equilibrium, it's a steady state. And so we talk about that as being treadmilling, that if you've reached that steady state where you're adding subunits at the front end as you're losing them at the back, in principle, you can kind of grow your way forward through that treadmilling process. This treadmilling is a logical consequence of this polar character to the filaments. It turns out that in the cell, the ends of the actin filaments are not free very often. And so you would think that it would be harder than for authentic treadmilling to happen in cells because you don't have the landing site, so to speak, for the monomers to land at. And in fact, the actin filaments are so important to cells that they have a wide variety of proteins that can interact with the filaments, doing different kinds of things with it. So for instance, you can have capping proteins that can cap either the fast or the slow end. There are different proteins there. And that locks it in, no more additions, no more losses. There are branching proteins where they can bind to the side of an actin filament and then grow a new one off the side. There are depolymerizing proteins that then catalyze the collapse of the filament. There are ones that can simply break it in the middle to sever it. And so all of these things will affect the dynamics of the actin filament in a living cell because the cellular context is many different proteins all present at the same time. One of the cellular behaviors that is extremely sensitive to actin polymerization is crawling. So if you're observing a fibroblast crawling along the surface, that behavior will stop quite quickly if you stop actin polymerization. Now, why would you care if cells are crawling? Well, there are a number of times that cells crawl that you're happy or sad that they do that. If we look during embryogenesis, there are multiple times when a group of cells is in one location and they have to crawl to reach another location where they're going to complete development. And so the muscle cells that are in our arms have crawled there out of somites earlier in development. This ability to move around is not completely lost, so that when you have a cut or something, as I mentioned, the fibroblasts and epithelial cells can literally crawl in to complete that wound and close it. A third time that cell movement is important, although not as desirable, is during metastasis. And so the initial tumor will be found somewhere in the body. If it finds a way to escape from that location into the bloodstream, it may metastasize and spread throughout the body. And elsewhere in the body, those cancer cells often have to crawl to find a home to make the next secondary tumors or whatever. Some cancer cells crawl to get out of the primary tumor. And there are genes that make cells crawl a lot that make them a more aggressive, more metastatic cancer. This is an example of what a crawling cell might look like. So you have a ruffled edge at the front of the cell and a tail where it's sort of attached. It has to let go there before it can get too far along. And inside of this ruffle is a huge number of microfilaments. So you can see in this electron micrograph just the density of the microfilaments. And this cell would be moving in this direction. So this is the free edge of the cell up there. Any questions about the actin filaments so far? Okay. Now, if all that actin did was polymerize, that would be cool anyway, but it's not. Actin can dynamically make things happen, contract or pull or whatever, and it does that because there is another protein involved called myosin, which is a motor protein. Actin and myosin were originally isolated from muscle cells, and they were called actomyosin at that time, meaning the stuff that makes muscles act. And then they discovered it's two proteins. Well, this is what myosin looks like. It's a much bigger protein than actin. It weighs about 5 times as much as actin does. But it has a property of being able to make a dimer with very interesting characteristics. So there is a long coiled part that makes sort of a spine to it. And hanging off of that spine are these 2 head groups that actually have the ATP binding and hydrolysis properties. In this area, right around there, there are some light chains that then modify the behavior of the actin molecules. Diagrams like this were happening for a long time just based on biochemistry. And more recently, we've been able to actually get pictures of actin molecules showing you that the diagrams, in fact, basically are what they look like. This molecule alone, under certain circumstances, does do some things. But what's even more important is the fact that this helical domain in here, this coil domain, allows it to form thick filaments. And so what you're seeing in this picture is an electron micrograph of many, many myosin molecules whose central tail regions have bound together to make a long cable, and their heads are sticking off the sides. So this is just sort of coated on the edges with these myosin heads. And it's the fact that this is a polymer of myosin that makes it possible for muscles to do what they do. And it looks something like this when we organize it. So if we want to look at the organization of it here, let's take a look in here. We have thin filaments or microfilaments that are organized parallel to each other and anchored off of a Z disc. And then we have thick filaments that are interleaved in between the thin filaments. They are centered on their bare region in the middle. And because of this mobile characteristic to the myosin heads, if in the relaxed state it's this long, if the myosin heads then walk along the thin filaments, the fiber as a whole contracts. The thick filaments are the same length that they were before. The thin filaments are the same length they were before. What's happened is they have moved relative to each other. So there's no actual contraction of any proteins that's happening during muscle contraction. The muscle contracts, the proteins don't. What makes that possible on a structural ground is the molecule that looks like this. So we have this neck region that we talked about that has places for light chains, a place to bind actin, and a place to bind ATP, and a place to bind actin. And as I just misspoke, then maybe that's a good time to reinforce what I said in my email message. If you see a typo in my handout as I'm talking about them, or if you hear me saying something wrong, please raise your hand and let me know. I would much rather correct things right away than after the fact, okay? I'm human. I know I can make mistakes. I try not to. I love to correct them when I can. So the way that works in practice is this. We have an actin filament. We have a myosin molecule with its neck and its head. And in this initial picture here, we're in what's called rigor state. The myosin is bound to the actin in a tight state. It gets its name from rigor mortis, when the bodies are rigid sufficiently after death. In this rigor state, if ATP is now added and it binds to that ATP site, now the head comes off of the actin filament. So adding ATP releases the actin filament. But like many proteins, myosin doesn't just hold on to ATP, it can hydrolyze it. And if it hydrolyzes it, then it changes the shape of the head relative to the neck. So hydrolyzing the ATP into ADP and inorganic phosphate flips that head forward like that. So that hydrolysis induces a change in conformation. That head can now rebind to an actin filament like this. And it's attached again to the actin filament, but at a different position than it was before. And the inorganic phosphate and PI can both diffuse away, and when they do that, it returns to its rigor state and bends back again. And so it's reaching forward and grabbing and just going forward like that. So it's a cycle. It's sometimes called a mechanochemical protein, as well as a muscle protein. And it's taking the energy of hydrolysis of ATP and turning that into movement, net movement. This myosin protein plays a role in a very large variety of cellular behaviors. So for instance, in cell division, it's myosin that pinches off the two daughter cells and separates them, so there's now two cells instead of one. It plays a role in cell movement. So in addition to the actin polymerization, there's a role for myosin in that kind of creepy crawliness we talked about. Obviously, muscle contraction is exquisitely dependent on it. And even the movement of organelles within the cell, so that, for instance, the pigment granules that give rise to color in skin and hair depend on a kind of myosin molecule to move around. Any questions on that before we move on? I think I don't know if we're going to have time to look at all these, but I'm going to look at the actin one at least, if we can pull this off. Okay. What on earth? Come on. This worked in my office. Maybe it'll work here. Maybe if we all petition the College of Medicine to put in faster computers. Here we go. Okay. So in this cell, what you're seeing is, in the green is actin that is labeled with green fluorescent protein. So this is looking at living cells where there is no separate dye. This is the protein itself that is visualizing the movement of actin during cell movement. And you can see how it gets concentrated at cell margins. You can see it rearranging during the process of movement. These are probably epithelials that are sort of moving as a sheet. You can see the periphery here of the ruffle. Right, one more time so you can see the that ruffling going on. Here's some ruffling going on. That's a dynamic process. And since this is in the link for this is in your handouts, you're certainly welcome to look at the other ones yourself. If we have time, we'll go back to the others also. So that's one big part of the cytoskeleton, the microfilaments, the microfilament system in myosin. The second major component of the cytoskeleton are microtubules, where microfilaments sort of feel like strings, if you like. They're fairly flexible if you have a long enough microfilament. Microtubules are tubules, and they tend to be more rigid. And they tend to resist bending. And so they're able to do what's called bare compression. Actin can bear tension when you pull on it. Microtubules bear compression when you push on it. So between these two systems, you're getting two of the important mechanical properties that you need. And what gives the basis for this rigidity that makes it possible for microtubules to do that? Well, tubulin is made of a dimer of alpha and beta tubulin. And it's always found as a dimer. You really can't get native alpha tubulin without beta tubulin. And they also bind nucleotide, but in this case they bind GTP. So you can see here GTP bound to each of those subunits. In the alpha subunit, the GTP is bound and it stays bound. It does not hydrolyze once the protein is synthesized. You can see perhaps why. It's kind of buried here underneath the beta. The beta tubulin, on the other hand, can hydrolyze its GTP to GDP, and it does so the same way that actin does after polymerization. So in both microfilaments and microtubules, making the polymer is coupled to hydrolyzing the nucleotide. It's not caused by it. You can use non-hydrolyzable analogs, and what you end up with are polymers that can't depolymerize very well. But if you look at this is again an asymmetric molecule. It has kind of a head and a tail to it, if you like. And as you might guess, the microtubule itself is also a polar structure. I'm using the diagram from your book since it's in there. I don't want you to carry away too much from it without my counteracting it. How's that for poisoning the well? Here we're looking at a mature microtubule that has 13 protofilaments. And in order to get to that point, a number of things have to happen. You can conceptually think of a protofilament of microtubules as being just a string of tubulins, head to tail, all pointing the same way. If you have a number of those protofilaments, you can sometimes find a sheet of protofilaments. And just as we saw with actin, the two ends of that are better or worse at binding the subunits. So one end polymerizes more quickly, the other end polymerizes less quickly. If you're under conditions in the test tube where you're making these sheets and so forth, the sides can come around and close off and make a complete microtubule like this. And the complete microtubule still has this polar character of having a rapid assembly end and a slow assembly or disassembly end. Doing this under conditions in the laboratory makes it possible to conceptually separate these out as distinguishable properties or characteristics. In practice, in the cell, it's more likely due to having gamma tubulin, which is a third kind of tubulin that's present in rings that are preformed, and then onto the gamma tubulin, the dimers then add to build out the microtubule. But in either case, there is still this polar character of having fast and slow assembly. You can further explore the mechanical properties or the structural properties of microtubules in the test tube by taking a whole bunch of them that are polymerized and now diluting them. You're lowering the concentration of monomers. Now you don't have enough to keep adding at the ends. What happens at that point when you can't keep adding? And in the test tube, what you can see are a sort of a fraying at the ends, a sort of a spraying out of the ends, which is a way of indicating that the side-to-side associations here are fairly strong, and that over time these end associations may let up, but you're still maintaining them in there. In the cells, just as for actin and microfilaments, both ends of the microtubules are probably interacting with something. And especially the slow assembly end is usually anchored to something. The fast assembly end is regulated in its behavior so that how fast it's going to polymerize will depend on the cellular conditions and so forth. Just as in the case of microfilaments, there are accessory proteins that can then interact with microtubules in functionally interesting and useful ways. So for instance, there are bundling proteins that will take, a number of microtubules and gather them together into a big bundle that's even stronger, as you can imagine, than the unbundled ones. There are others that are more loose and floppy like this and can cross-link between them and end up making more of a airy, low-density structure, more maybe like a geodesic dome. Certainly one of the key places that microtubules play a role is in cell division. It's microtubules that are used to separate the chromosomes at mitosis. And for that reason, drugs that target the microtubules are anti-cancer agents because they target cell division. Vinblastine and Taxol are drugs that bind to and stabilize polymers. The Taxol stabilizes it in this form of a microtubule. Vinblastine stabilizes it in crystals so that there isn't free monomer around to polymerize into microtubules. This is showing you a couple of these anti-microtubule drugs. And I think if you kind of look at it cross-eyed, maybe, you can see places where it suggests a similarity to the structure of GTP. So one possibility is in some cases it's binding at the GTP binding site and interfering with its normal behavior. Now, just like microfilaments, microtubules also have motor proteins that are associated with them, but these are different motor proteins. We've already mentioned that chromosomes can move along microtubules, and this is a particularly beautiful picture of those that happening. You have here an aster forming at the time of mitosis with probably the aster itself indicated by red. It might be G-tubulin, I don't know for sure. The green microtubules, and then the blue of the chromosomes, probably just stained for DNA. And then you can see how they're anchored onto and are getting ready to move apart because of the microtubules. Just as myosin had a direction of movement, the microtubule motors also have a direction of movement that depends on which motor you're talking about. This particular motor here is kinesin, and it looks somewhat similar to myosin, but it has some important differences. It also has a long tail that's a coiled coil. Its light chains are at the tail instead of on the neck, which is kind of kinky. And the light chains at the end there are part of how the cell decides what cargo the microtubule motors are going to carry on microtubules. So the kinesin is a very major player in moving particles around the cell. Here we just see that in a little bit more detail. Here you can see that as ribbon diagrams. You can get a sense, I hope, of the coiled tail there and the compact domains. And this is showing you the behavior of kinesin schematically, that it will overall move a cargo of some kind, a particle, a vesicle, whatever, from the minus end towards the plus end of the microtubule. The way that kinesin works is a little different from myosin. Myosin had a very rigid neck. It really had kind of two states it tended to be in, and they were quite precise. And so let's take a little look at that. If you have a head that has no ATP on it, it will be bound to the microtubule. So that's somewhat similar to the myosin case. When that head binds ATP, it induces a change in shape. And the change is to sort of sling the other head forward. So as this attached head binds its ATP, the other one is now whipped around towards the plus end. When that second head attaches to the microtubule, it will release its ADP. And now it has a firm attachment, and it communicates that to the back end where it can now hydrolyze the ATP to ADP, and now we're set up to do all of that over again. So what's interesting here is that you always have somebody who's got a foot on the microtubule. The first foot, and then you swing around, and then you swing around. So you're always attached to that microtubule. And a consequence of that continuing attachment is that we call the motion processive. That is, this is an enzyme that can do it again and again and again and again and again. We're going to see some other very important enzymes that are processive. For most enzymes, it's a hit or miss thing. You come, you do it, you go away. Processive enzymes always have something interesting going on, and in this case, this is one of the things that, for instance, can get vesicles to the very end of neurons. You know, I've got a neuron that starts here and goes down to my fingertip. It's a good thing that kinesin is a pretty processive enzyme to get those vesicles down there. But we do still, again, have ATP being used. We do still have a change in conformation being involved, and a hydrolysis of it. Well, the third system we have are the intermediate filaments. These are much, much more diverse than actin or tubulin, and I'm not going to try to go through all the major families. That's a lecture in its own right. Ones that are in the skin are a family called keratins, and they're present in the skin and in other epithelia. We've been able to see, We've been able to see that intermediate filaments play an extremely structural role. They resist mechanical stress. Actin filaments can generate tension using myosin. Thick intermediate filaments are just kind of good at passively resisting it. The names of these filaments, by the way, come from their diameters. Thin filaments are the thinnest of the three. Thick filaments are the thickest of the three. And intermediate filaments are intermediate in thickness between the myosin and the actin filaments. Intermediate filaments, unlike microtubules or microfilaments, are not polar. There is no difference between the two ends, and we're going to see why structurally in a second. There is no place on intermediate filaments to bind nucleotides. So although actin binds ATP and tubulin binds GTP, intermediate filament proteins don't bind anything. And the way they form a polymer is to form a coiled coil. And we're going to take a little look at that structure because coiled coils turn up in a lot of different contexts. Intermediate filaments can interact with microfilaments and microtubules by cross-linking proteins. And so more and more over time, we're finding proteins that specifically cross-link intermediate filaments into the other two cytoskeletal systems. Coiled coils are extremely common as a structural motif. You actually saw them in the myosin tail and in the kinesin tail. But it was in the intermediate filaments that they were probably first and best understood. If you think about the alpha helix, as you go around that helix, there's a repeating pattern. And if you, on the first and fourth positions, tend to have hydrophobic side chains, and the partner tends to have hydrophobic side chains at the first and fourth positions, then there's going to be this oily surface along the alpha helix. And so two alpha helices can coil around each other, and that's why we call it a coiled coil. Each one individually is a coiled polypeptide, making an alpha helix. Two of them together coil together to make the coiled coil. We have here three representations of it that bring out different aspects of it. Here's just the alpha carbons, and I think you can see there the twistiness around each other fairly well. Here you can see the side chain sticking out and the way you have contacts in the middle and the side chains coming off. And then in the space-filling diagram is probably the clearest of all. This is a left-handed helix. The alpha helix is a right-handed helix. The coiled coil is a left-handed helix. It takes two right-handed helices and coils it in the left-handed version. So up until this point, you might have been forgiven for thinking that all biological systems are right-handed, but zDNA is left-handed and the coiled coil is left-handed. Any questions about this so far? Okay. So how did we get there? What gives rise to these structures that we see in the epithelium and so forth? You can think about a single monomer, a single polypeptide of one of these intermediate filament proteins. You won't find it free in solution. These are not very soluble proteins, but you can certainly imagine one, thinking of it as it's been translated. Two of those monomers make a dimer or a coiled coil. And if you look at this dimer, you'll notice that it's in this, it itself is polar. It's got two little red dots at one end and two black dots at the other, meaning that they're being coiled around each other in the same direction. The N terminus and C terminus, the two N termini at one end, the two C termini are at the other end. But the reason that the filament as a whole is not polar is because these dimers then become staggered in the tetramer and packed together. So overall, the intermediate filament has no net polarity. You can see here, this one's pointing red this way, that one's pointing red that way. So you end up packing them together like bricks in a wall. And because there's equal numbers pointing both directions, there's no polarity to the filament as a whole. Extremely insoluble, among the most insoluble components in the cell. Since they don't bind nucleotides, they don't need to hydrolyze them, and the polymerization from monomer to full intermediate filament can occur completely spontaneously. You can take a filament like this and solubilize it in urea, dialyze out the urea, and reform intermediate filaments with nothing else providing information. It's so insoluble, in fact, that you might wonder how the cell can make something that's that insoluble and have it be in the right place. We know for vimentin, at least, which is one of the intermediate filaments, that some of it is assembling as it's being translated. That there's some of it that is in structure as a nascent polypeptide. And as you might expect, the messenger RNA for it also shows some patterns of localization. And that if you were to mislocalize its mRNA, it then distorts the structure of the filaments. Let's move outside the cell. We've talked about 3 cytoskeletal families inside the cell, certainly a large fraction of the structural proteins. But there's also a big chunk of them outside of the cells, and the most conspicuous of those are collagen. Collagen means glue maker, and you can use collagen to make glue. That's the original glue. Collagen is a very strange protein in that it's not just rich in proline, which is usually a rare protein. Most proteins don't have more than a few percent proline. But they also have a lot of hydroxyproline. So you have this proline ring, and after synthesis, a hydroxyl group has been added to it. So this is normally rare, this is a very unusual amino acid, and yet they're very abundantly present in the collagen molecule. The collagen has a conformation all of its own. It has a secondary structure that's just about unique, perhaps unique to collagen. Do you remember, Susan, if it's unique to collagen? Yeah. So collagen alone has a secondary structure of a left-handed helix. If you look at this left-handed helix, it is a much more open, extended helix than the alpha helix. So the left-handed collagen helix is quite different from an alpha helix, but it's still a form of secondary structure. It's a local repeating order. If you have 3 molecules of collagen with their left-handed helix, they can make a coiled coil, but this is a triple coiled coil. So 3 strands of collagen together can twine around each other, and then it turns into a right-handed coiled coil. So in the case of intermediate filaments and myosin tail and the kinesin tail, the right-handed alpha helices made left-handed coiled coils. Collagen has a left-handed secondary structure that coils up into a right-handed coil in its tertiary structure, in quaternary structure, I should say, because this is multiple peptides. Each one of these collagen molecules then can side associate with other collagen molecules until you end up with very large bundles. And you can see from the little bands that it's a repeating structure. The little staining there is to emphasize that in that coil there's a very, very regular and repeating pattern. When we look at that coiled coil, that triple coil, we see why it's so important to have the sequence or the composition it does. An awful large fraction of the sequence of collagen is simply glycine, proline, hydroxyproline, and repeat it till you're bored. So again and again and again and again. That by itself is capable of making this extended left-handed helix, but when you pack them together, the glycines can fit in the middle of that triple helix. Only glycine is small enough to fit in there. Also, it's kind of kinky, you notice there. And so you have at the core of the collagen triple coiled coil this solid centerpiece of the glycine molecules. And then the proline and hydroxyproline are sticking out to the sides here, ready to interact with other molecules of collagen. Collagen is even more robust than that because among the few side chains that aren't glycine, proline, or hydroxyproline, there can be covalent cross-links. So there's a fair bit of lysine in collagen, and that lysine can actually be cross-linked with a covalent bond to end up looking like this. It's this cross-link that requires the presence of ascorbic acid, and the reason scurvy happens is that you can't cross-link your collagen, and so it's too soft and tends to fall apart. Yep, there we go. Very quick side note, the collagen in your body is being remade and replaced all the time, including in scars. And so if you are vitamin C deficient, the first collagen that shows the deficiency is the collagen in the scars because it's less well organized than the rest of your body. And so one of the tragic things that can happen is that old wounds can reopen in people who have scurvy. And this is what happened to Titus Oates when they were going to the South Pole. Have a great weekend. Sorry, what?